attendance. So according to attendance here, we see 1 hour. Okay, Muhammad Hassan, I'm glad that you are joined with Facebook here. Snapshot. Okay, so today's topic is apoptosis. We'll start with apoptosis. Is the pathway of cell death in which cell activate enzymes and degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. Fragments of the apoptotic cells then break off, giving the appearance that is responsible for the name apoptosis, also known as falling off. So the plasma membrane of the apoptotic cell remain intact. The membrane is altered in such a way that the fragments called apoptotic bodies become highly edible, leading to the rapid consumption by phagocytes. So that cell and its fragments are cleared with a little leakage of cellular contents. So apoptotic cell death does not elicit an inflammatory reaction. Thus, apoptotic differs in many respects from necrosis. So that what so in easy way, so apoptosis is the best way of cell death. This cell death doesn't is not followed with damage of different tissues, is not followed with any kind of inflammation, and so yes, really the best way of this the best way of pathology to clear their consistence from cells. So, causes of apoptosis. Apoptosis occurs in many normal situations and serves to eliminate potentially harmful cells and cells that have outlived their usefulness. It also occurs as a pathologic event when cells are damaged, especially when the damage affects the cell's DNA or proteins, thus the irreparably damaged cell is eliminated. So let's okay, in which conditions we have this situation? Later we'll discuss in more details. During embryogenesis. So during embryogenesis, so a lot of cells which are useless. They're useful during the embryogenesis, but after the embryogenesis is over, so no more stimulation by growth factor happened, and that's why the cells without supportion of growth factor signaling, so they died. Then proliferative tissue turnover, loss of growth factor signaling to some tissues, for example, intestinal epithelium, lymphocytes, in bone marrow, and thymus. They gradually leads to death of the cells. By apoptosis without inflammation. I repeat it again. It's normal physiologic condition. It's this pathway is necessary to elicit the amount of cells which shouldn't be present in this site. Then involution or hormone dependent tissues. So along with growth factors, we have hormone stimulating cells. So usually it's our, for example, cells of our genital tract. I mean genital organs. They are hormone dependent, and if we lose the hormonal intake, so a lot of these tissues get degraded. So involution hormone dependent tissues. Example is endometrium. So every menstrual cycle is controlled by estrogen progesterone levels, and when progesterone levels drops in secretory phase, so endometrium sheds out with the bleeding. Yeah, this is typical example apoptosis. Then, decline of leukocyte numbers at the end of immune and inflammatory responses. So when we have inflammation, so too many cells are recruited to this area, and then our task is to diminish the amount of these cells. It also responsible. It's also handled by decreased. Sorry. By decreased amounts of hormone signaling, not hormone, but growth factors, hormone, and other receptor-like signaling to the leukocytes. Elimination of potentially harmful self-reactive lymphocytes. So when self-reactive lymphocytes are there, it leads to it leads to diminishing of the population of this lymphocytes, which they can potentially can damage our own cells. So that's why we need it. Then in pathologic cases, so it's also best response in pathologic cases because some cells during their life they undergo the DNA damage. And in case of DNA damage, we have specific mechanism which we are going to discuss today. When the activation of pro-apoptotic proteins goes, so by BH3 only sensors. What is BH3 only sensors? We will discuss today. Accumulation of misfolded proteins. So when proteins are misfolded in the plasma reticulum, I said about it in a few lectures ago, that when misfolded proteins are collected, so same BH3 only sensors are activated, and they activate the apoptosis, which is related to caspases later. And infections, some infections, especially viral infection, they also cause the changes in cell metabolism, and our cells decide to kill themselves. Instead of treating of cells. We just kill these infected cells, and it sometimes it leads to elimination of virus from the cell, but also it explains the pathogenesis of a lot of viral diseases. For example, so one of examples is okay, our coronavirus. So it entered into our cells and facilitate their function. So they do their function. They produce. This virus is spread out and enter in every cell. And then when our immunity cells, so cytotoxic T lymphocytes, start to kill the cells, of lungs, and instead of this dead cells, produced the connective tissue, scar tissue, and it is known as fibrosis. Yeah, so you know that one of the main outcomes of so bad outcomes, complications of the coronavirus is fibrosis of lung. It happened also with help of apoptosis. So because T lymphocytes killing the cytotoxic, killing these infected cells. But if there are most of cells damaged by virus, infected by virus, so we have nothing to replace them, and that's why we have production of connective tissue there. Okay, it's in next lecture about the tissue injury. We'll discuss it in details and how healing happened in these cases. Okay. Apoptosis in pathologic condition and physiologic apoptosis is discussed. Let's start to talk about a mechanism of apoptosis. Mechanism of apoptosis is divided into two types. It is intrinsic pathway, also known as mitochondrial pathway of apoptosis, and extrinsic pathway, so it's a cytotoxic pathway. But crucial and main point of this that all the mechanisms are come to one space, to one enzyme known as caspases. Caspases are cytoplasmic enzymes which provide no function in normal cell life, but they are it's like suicide pills for the cell, and in any moment, so it can cause the inflammation. Okay, so yeah, it can cause the inflammation. Cause the death. Sorry, it can cause the death. Sorry. Oh my bad. So caspases were so named because they are cysteine proteases. They cleave proteins after aspartic and acid residues. Two distinct pathways converge in caspase activation: the mitochondrial pathway and the death receptor pathway. So, but finally, both of them will come to caspases. Let's talk about this mechanism. So, mitochondrial intrinsic pathway seems to be responsible for apoptosis in most physiologic and pathologic situations. Mitochondria contain several proteins that are capable of inducing apoptosis, including cytochrome C. You remember in explanation of mitochondria in a few lectures before, I explained that inside the mitochondria we have cytochrome C protein, and it contains inside the mitochondria and provides some specific biochemical function inside. But along with it, the cytochrome C, so when it's leaked outside the cell, it provokes the apoptotic events. So take a look here. This is mitochondria. So inside contains the cytochrome C. And you remember that in case of cell damage, so somehow, by which process I'll explain later, open the channels here, channels in mitochondria, and cytochrome C leaks out. And cytochrome C, with help of regulators, okay, let's discuss. During the cell injury, so might be growth factor withdrawal, so decrease of growth factor intake, might be DNA damage. It's innate cause, and protein misfolding happened. So you remember endoplasmic reticulum stress. All of them activate the BH3 sensors. BH3 sensors are also proteins inside the cytoplasm, which later activate the BCL2 family effectors, which are known as BAX and BAC. And they affect mitochondria and with help of regulators BCL2 and BCL-XL, channels which can transport cytochrome C outside the mitochondria entered inside the cytoplasm, and it causes the activation of caspases. And caspases, there are proteins in the cytoplasm which form the apoptotic bodies and causing the first nuclear fragmentation, and it's one effect of caspases. And second event is breakdown of cytoskeleton. When cytoskeleton is broken down, so blebbing of the cell membrane goes very fast. Blebbing of cytoplasm goes very fast, and more and more cytoplasm get outpoached from the cell, with organelles inside, with residues of cell membranous organelles inside, and forming the apoptotic body. And these apoptotic bodies are produce specific ligands for phagocytic cell reception. We have phagocytes there inside the tissues, which will destroy these apoptotic bodies. So entire cell finally will split into these apoptotic bodies, and these apoptotic bodies, without any inflammatory reaction, is phagocytosed by macrophages. It might be macrophage, it might be the monocytes if we're talking about the blood. But anyway, this structure is destroyed. Okay, let's talk about details. Yeah, here I explained. Then the death receptor pathway, so it's extrinsic pathway of apoptosis, is related to this simple descending pathway. So we have special receptor ligand interactions. Then we have FAS receptors and TNF receptors. TNF receptor is tumor necrosis factor receptor. So our cells in immunity will discuss it, but some cells of our immunity they produce TNF, tumor necrosis factor, and this tumor necrosis factor activates the receptors of tumor necrosis factor. Then we have some adapter protein. You remember it from previous lectures. Now we have adapter proteins which are produced like transmits the signal to caspases, and same mechanism as cytochrome C activates the caspase reaction, and adapter proteins activate the caspase reaction, and outcomes after this goes absolutely similar way. We have activation of endonucleases. Endonucleases are enzymes which will destroy the nucleus and chromatin. And chromatin is fragmented, skeleton of nucleus was destroyed. Cell membrane, nuclear membrane, forming also blebs. So these vesicles filled with residues of chromatin and DNA, and forming these small vesicles, which will also later will income into the apoptotic body, and then this apoptotic body is swallowed by phagocytes. You see, similar, absolutely similar thing, but here it comes because of innate reasons, some pathologic reasons outside the cell. But here we have another process when receptors are activized and producing also caspase activation. So you see, crucial thing for this is the caspase activation. Okay. Clearance of apoptotic cells. Apoptotic cells and their fragments entice phagocytes by producing a number of eat-me signals. We have eat-me signals on the surface of apoptotic bodies. For instance, in normal cells, phosphatidylserine is present on the inner leaflet of the plasma membrane, but in apoptotic cells, this phospholipid flips to the outer leaflet where it is recognized by tissue macrophages, leading to phagocytosis of the apoptotic cells. So you see, this is in apoptotic cells we have specialized signal. It's once again, sorry. Okay, let's continue. Leaflet of plasma membrane, but in apoptotic cells, this phospholipid flips to the outer leaflet and recognized by macrophages a special signal which leads to activation of macrophages only, not inflammatory reaction, but macrophages only. So and macrophages will clear this. Okay, in histological examination of apoptotic bodies, we can hardly find this apoptotic cells. Very hardly we can find it because the apoptosis is never outnumbered. Apoptosis always goes very gradually in a very small speed, and in histological examinations, very hard to detect the cells. Cells rapidly shrink, form cytoplasmic buds, and fragment into apoptotic bodies that are composed of membrane-bound pieces of cytosol and organelles, as I shown before. Because these fragments are quickly extruded and phagocytosed without eliciting an inflammatory response, even substantial apoptosis may be histologically undetectable. That's what I said to you. Then let's talk about other pathways of cell death. In addition to necrosis, apoptosis, two other patterns of cell death have been described that have unusual features. Also, the importance of these pathways and disease remains to be established. They are the subjects of considerable current research, and it is useful to be aware of the basic concepts. You see, we have necroptosis. Necroptosis is, you see, it's composed of two words, necrosis and apoptosis. Necroptosis, its form of cell death is initiated by engagement of TNF receptors as well as others poorly defined triggers. Unlike the extrinsic pathway of apoptosis, which also is downstream by TNF receptors, as I shown before, in necroptosis, kinases called receptor interacting protein (RIP). Kinases are activated, initiating a series of events that result in the dissolution of the cell, much like necrosis. So you see, stimulation is similar, but outcome is a little different, just because of different outcome. Um, and name came from necrosis and apoptosis. In some infections, this specific pathway, so it's like fusion of two pathways of cell elicitation, present in some infections. And it has been hypothesized to play a role in ischemic injury and other pathologic situations, especially those associated with inflammatory reactions in which the cytokine TNF is produced. TNF, once again, is tumor necrosis factor. When and why it occurs and how significant it is in human disease is not well understood. But just keep in mind, we have necroptosis. It's also one way of cell death. Pyroptosis. Pyroptosis, this form of cell death, is associated with activation of a cytosolic danger sensing protein complex called the inflammasome. So this pyro means heat, yeah. So it happened in heat condition. Okay, I'll show it to you. The net result of inflammasome activation is the activation of caspase, some of which induce the production of cytokines that induce inflammation. So you see, it goes like apoptosis, but with inducing of inflammation. So it's manifested by fever and other trigger apoptosis. Apoptosis and inflammation coexist. You remember I said to you that apoptosis, main feature of apoptosis, it is not followed with inflammation. But this is mechanism of this is very resembles the apoptosis. So same caspase activation, but along with it, it causes the inflammation. So some microbes cause the death of infected cells. Its role in other pathologic situations is unknown. Autophagy. Autophagy is also a type of cell death. Autophagy, it's not cell death actually itself. Sometimes autophagy is a mechanism by which we digest our excessive particles inside the cell. So we have lysosomes. You remember I explained you lysosome function, and lysosome used to be known as the organelle which digests the particles inside digests the phagosome. Yeah, so it fuses with phagosome and forming the phagolysosome, by which we can destroy ingested material. But also the same idea is possible for our own cells inside organelles, our own cell organelles. For example, all life. For example, some organelle in our cell, for example, mitochondria, is damaged. Yeah. So we have deprivation of nutrients, and we try to find something to eat. Yeah, and we destroy some organelles inside our cells to destroy them into the building blocks. Might be amino acids or to lipids, anti-lipids or carbohydrates, to use them for another more important today reaction. I can compare it with so actually it's related. So when you are active man, yeah. So when you are going to gym, you pumping iron, yeah. Your muscles are very huge, and then you're very attractive man, yeah. But then you stop doing gym exercises. You don't need them, and you stop doing this, and then you became starving. You can't eat. You have no food, yeah. And then what your body decides to do? It starts to destroy your muscles. So gradually destroy your muscles to produce more amino acids just to survive. And similar process goes inside on the cellular level. Then inside the cell, some organelles are destroyed and some some cell parties are destroyed. And this mechanism is necessary to survive the dying cells. So also due to insufficiency of feeding. Okay, and later gradually it will come to, so this mechanism is explained. So some cytoplasmic organelles get surrounded by autophagic vacuole. Then this autophagic vacuole fuses with lysosome, and then so when these organelles are destroyed, it is used as source of nutrients. So this mechanism is not only pathologic; it also happens in normal situation. So sound off, please. It also happens in normal situations when we just renew our organelles. So now all our organelles, all our cell membranes, they have charge to be starved. So you remember in some previous lecture I said that mitochondria is trained, yeah, and lives half-life of mitochondria is approximately 10 days. And after 10 days, what happened with them? After 10 days, mitochondria get destroyed by their own lysosomes, by cells' own lysosomes. And we use the fragments of old mitochondria to build up a new mitochondria as a building block in normal condition. In pathologic condition, of course, it leads to atrophy, so cell atrophy. Then summary. Let's talk about apoptosis and necrosis summary. Then we'll switch to mechanisms of cell injury and cell death. Reversible cell injury. It causes cell swelling, fatty change, plasma membrane bleeding, and loss of microvilli. Mitochondrial swelling, dilation of the ER, eosinophilia, resulting from decreased cytoplasmic RNA, and also causing by denaturation of proteins. I explained you in details in previous lecture. If you didn't capture it, so please open the link I shared you in YouTube to observe this lecture again. Then necrosis. It is accidental cell death manifested by increased cytoplasmic eosinophilia, causing the nuclear shrinkage, fragmentation, and dissolution. It also causes the breakdown of plasma membrane and organellar membranes, abundant myelin figures, leakage of enzymatic digestion of cellular contents. Then morphologic types of tissue necrosis under different conditions. Necrosis in tissues may assume specific patterns. It might be coagulative, liquefactive, gangrenous, caseous, fat necrosis, and fibrinoid. All these types are discussed in previous lecture. Then apoptosis, part of our lecture, regulated mechanism of cell death that serves to eliminate unwanted and irreparably damaged cells with the least possible host reaction, characterized by enzymatic degradation of proteins and DNA initiated by caspases, and by rapid recognition and removal of dead cells by phagocytes. Apoptosis is initiated by two major pathways. It's mitochondrial and death receptor pathway. So it's also named as intrinsic or extrinsic pathway. So keep it in mind. Intrinsic pathway happened due to activation of mitochondria. So it's triggered by DNA damage, so ER stress, or any other mild damage of the cell, so which leads to activation of BH3 only BH3 only receptors, so which activates itself the BCL2 family of proteins, which triggers the mitochondria to open channels for cytochrome C, and then cytochrome C activates the caspases. And after this, caspases are destroying the cell from inside. And we have death receptors, which with help of specific receptors like TNF receptor. In TNF receptors are activated usually by cytotoxic lymphocytes, cytotoxic T lymphocytes. So cytotoxic T lymphocytes are here. This is abbreviation CTLS. And then TNF receptor family directly activates the caspases. and then activation of caspases leads to cell death. Then we have two other unusual pathways of cell death, which are also related to apoptosis. It is known as necroptosis. It has the signs of necrosis and apoptosis both. And also pyroptosis, which is apoptosis with inflammatory reactions. And autophagy, it's not related to apoptosis at all, but it doesn't lead to cell death. It's adaptation to nutrient deprivation, in which cells digest their own organelles and recycle them to provide energy and substances. If the stress is too severe for the process to cope with it, it results in cell death by apoptosis. So this autophagy is beginning of apoptosis, so pre-apoptotic changes inside some cells. Then let's talk about mechanisms of cell injury and cell death. The cell response to injurious stimuli depends on the type of injury, its duration and severity. Low dose of toxins or a brief period of ischemia may lead to reversible cell injury, but larger toxins doesn't, or longer ischemic times may result in irreversible cell injury. And of course, the reversible cell injury is leading to cell death. Consequences of an injurious stimulus also depend on the type, status, adaptability, and genetic makeup of the injured cells. So, for example, so here it's explained, I'll explain it in an easier way. For example, our brain cells, they are very dependent on our nourishment of oxygen, of amount of glucose level, and even 20-minute deprivation of oxygen level so might lead to death of neurons. And that's why when heart stops, your main task to keep pumping his heart, to give the blood, to provide the blood into the brain. That's the main idea of ABC. So now it's CAB actually, cardiovascular resuscitation. Is okay. But some other tissues, for example, bone cells, they can survive even months after the death. Because they don't need a lot of nutrition. They can survive by eating tissues around, and that's why they live for much longer. Same to muscles. Yeah, so that's why you heard the situation when a patient has died, but then we adjust electrodes to the muscle, and muscles will keep contracting. They'll keep contracting, and that's these studies are started from when we open the electricity, and by electricity we can trigger the muscle contraction even after death, after brain death. The first tissue which is died after the injury, it is brain, of course. Then cell injury usually results from functional biochemical abnormalities in one or more or limited number of essential cellular components. As we discuss in more detail later, different external insults and endogenous perturbation typically affect different cellular organelles and biochemical pathways. Deprivation of oxygen and nutrients, as in hypoxia and ischemia, primarily impairs energy-dependent cellular functions, culminating in necrosis, whereas damage to proteins and DNA triggers apoptosis. You see, deprivation of energy causing the necrosis, and damage to proteins and DNA triggers apoptosis. That's main differences. Okay, it should be emphasized that the very same injurious agent may trigger multiple and overlapping biochemical pathways. That's why we can't totally say that here will be necrosis, here will be apoptosis, because one same injurious stimuli might cause both the necrosis and apoptosis. You see, as it's shown on diagram, deprivation of ATP causing the decreasing of ATP synthesis function, and all energy-dependent functions are shut down. It causes the cell injury. And due to multiple injurious stimuli, so increase the amount of radical oxygen species, which we also will discuss in this lecture a little later. It causes the damage to lipids, proteins, nucleic acids, and also causes the cell injury, which leads to necrosis. Then But mutations, cell stress, infections, they cause the wrong production of proteins, and proteins became misfolded, and it causes the ER stress. BH3 only family which detects these misfolded proteins, they activate the cascade of changes which activate the intrinsic pathway of apoptosis, and caspases. When caspases are activated, apoptosis is in process. Then, for example, in radiation and other insults to DNA, DNA has been damaged, and nucleus reaction. Here we have p53 family proteins, which also activize same BH3 family proteins, which later leads to apoptosis. Um, but infections, immunologic disorders, they have a huge variety of types of injury that might produce toxic molecules, which might lead to or necrosis or apoptosis. So, especially in inflammation, we can find both necrosis, apoptosis, necroptosis, and pyroptosis. So, anyway, it will lead to death. Let's talk about injury of hypoxia and ischemia. Deficiency of oxygen leads to failure of many energy-dependent metabolic pathways and to death of cell by necrosis. Most cellular ATP is produced from adenosine diphosphate by oxidative phosphorylation during the reduction of oxygen in the electron transport system of mitochondria. High energy phosphate in the form of ATP is required for membrane transport, protein synthesis, lipogenesis, and the deacylation reacylation reactions necessary for phospholipid turnover. And keep it in mind that all these processes are life-threatening. So, and first is membrane transport. So first of all, membrane transport suffers. So no proper sodium potassium pump works there. No more sodium expelling from the cell happened, and entrance of potassium happened inside the cell, and resting potential is decreased in the cells. And more water intake to the cell going on. That's a first damaging point. So that's why cells are swelled during the hypoxia and ischemia. Then protein synthesis is disrupted. So our cells can produce proper proteins to sustain the life of the cell. That's also one of ideas. So why cell death happened. Then lipogenesis, no more membranes happened, and as you know, so membranes of cells they have very short lifespan, and we need to renew them very fast. And then when cell membrane so uh, um, is deprived from lipids, so it's not renewed. It became root, it became solid, and due to this, it's very easy to rupture this type of cell membrane, and cell membrane rupturing directly will come to cell death. Also, we have a very important process, deacylation and reacylation. So adding of acyl radical to some components. It's also in lipid formation we use deacylation and reacylation, and in a lot of different reactions it's necessary, especially in phospholipid turnover, as I said. So that's why ATP causes these outcomes. So hypoxia-inducible factors. But in some tissues, cells subjected to the stress of hypoxia that do not immediately die activate compensatory mechanisms. We have compensatory mechanisms, and they are named as HIF, hypoxia-inducible factor 1. This hypoxia-inducible factor 1 stimulates the synthesis of several proteins that help the cell to survive in the face of low oxygen. Some of these proteins, such as vascular endothelial growth factor, VEGF, which we discussed a few lectures before, stimulate the growth of new vessels in this tissue, so to facilitate a larger amount of blood vessel in this area. And that's why in areas of ischemia, you will find a lot of blood vessels which going around this necrotic space. So that's in histological observation. I will show it to you. When so necrosis of heart tissue happened due to ischemia. So we will find a lot of blood vessels around this necrosis. That's how our tissue tries to avoid hypoxia, but it works poorly. Other proteins induced by HIF1 cause adaptive changes in cellular metabolism by stimulating the uptake of glucose and glycolysis and dampening mitochondrial oxidative phosphorylation. And then anaerobic glycolysis can generate ATP in the absence of oxygen using glucose derived either from the circulation or from the hydrolysis of intracellular glycogen. So I remind you, we have another pathway of ATP production. It's anaerobic pathway. So without using of mitochondria, without using of oxygen. And in acute situations, especially in muscles, we can go to this. We use our glycogen storage, and due to this glycogen storage, we reactivize it. And ATPs are produced. Yes, less amount, but in a life-threatening situation for the cells, we can use this ATP. That's why tissues with a larger amount of glycogen can survive the hypoxia better than tissues without it. And examples here are muscles, skeletal muscles. Also not only skeletal muscle but heart muscle too. Also muscles of also present in liver, so glycogen storage, and due to this they can survive the ischemia for a longer time. But negative outcome of this is that actually more that produced a lot of acids, and these acids they coming to produce acidic milieu inside the cell, which also can damage this cell. So that's in pathology of cardiovascular system. We'll discuss the ischemic injury of the heart, and this process helps to survive more, but later it comes to some severe effects. So especially, as I want to remind you that in hypoxia and ischemia injury, so long-term ischemia causes the usually coagulative infarction, coagulative necrosis. When high, acidic pH inside the cell will cause the denaturation of proteins, and it happens the same thing when you boil eggs. Then liquid intracellular material became solid and white in color. So that is the outcome of this anaerobic glycolysis. But sometimes this aerobic glycolysis is skipped by process known as Warburg effect. Warburg effect I little explained to you in previous lectures. It's some cells consciously come to the anaerobic glycolysis and skip the mitochondrial activation and produce building blocks for other cells. Especially it's necessary for cells, fast renewing cells. For example, cells of skin, epidermis of skin, might be and especially in tumor cells. And tumor cells start to use this Warburg effect. They don't need oxygen too much. They need oxygen just in little amounts. So some tissues, not all blood, not all cancer cells, but some cancer cells, they undergo Warburg effect, and they produce more amino acids. They produce more nucleic acids, and finally it leads to growth faster. They need more intake of nourishment but less intake of oxygen. Okay, this we'll discuss a little later in a few after a few lectures after this. And persistent or severe hypoxia and ischemia ultimately lead to a failure of ATP generation and depletion of ATP in cells. So that's what I said to you about. ATP-dependent sodium pumps are reduced in activity, which causing the accumulation of sodium and efflux of potassium inside the cell. The net gain of solute is accompanied by isoosmotic gain of water, causing cell swelling and dilation of the ER. I explained this to you, so it shouldn't be new for you. The compensatory increase in anaerobic glycolysis leads to lactic acid accumulation, decreased intracellular pH, and decreased activity of many cellular enzymes. So that's why I said this is another surface of the same coin. Yeah, that actually intracellular pH increasing due to this. It helps a little bit to produce ATP, but also due to these enzymes stop working. It also might lead to cell death. Prolonged or worsening depletion of ATP causes structural disruptions of the protein synthetic apparatus, and that's why RER detaches the ribosome from its surface, and polysomes are dissociated, and protein synthesis is stopped, is dampened. And then we have that hypoxia per se increases the accumulation of radical oxygen species. So it's counterintuitive, but deprivation of oxygen causing the increasing of reactive oxygen species. We'll discuss it a little later, but also very interesting phenomenon known as reperfusion injury. So. And we have ultimately there is reversible damage to mitochondrial and lysosomal membranes, and the cell undergoes necrosis. Membrane damage is a late event in cell injury caused by diverse mechanisms and is discussed later. Then ischemia reperfusion theory. So what is time now? Yeah, actually this point I'll explain to you next lecture. So now let's check the attendance again. Yes, I've checked. The Hyprabi also is here. Then thank you for attention. I hope you enjoyed this lecture. See you next time.
Okay, sir. Bye bye.
Okay, sir. До свидания. Пока-пока.
Thank you.
Okay, sir. Take care.
Any questions?
Have a nice day.
Thank you, sir.
Sir, can we leave?
This video will be uploaded into the YouTube.
Thank you, sir. Okay, sir.
Okay, sir.